This is a presentation on the use of the polymerase chain reaction in science, as narrated by Andre, Alice, and Jade. It will cover content on the history and development of the biological technique, how the polymerase chain reaction works, the current applications of the technique, advantages and disadvantages of the technique, and possible future applications of the polymerase chain reaction.
Polymerase chain reaction, or commonly referred to as PCR, is a molecular biology technique developed by Kari B. Mullis in 1983, which is used to amplify or reproduce particular sections of DNA or RNA. One can start with a single molecule of DNA, and the PCR can generate 100 billion molecules in a short period of time, as each copy will increase exponentially. It is a relatively easy process to perform, as it only requires a test tube, heat and simple reagents, and a DNA sample. The sample doesn't need to be uncontaminated or new to be successful in the replication, as PCR has worked successfully on DNA from tissue samples, human hair, dried blood, and even from tissues of a mummified brain. PCR is widely used in gene analysis, along with being a diagnostic tool for genetic diseases, the detection of bacterial, viral and fungal pathogens, and it has a potential in forensic medicine. There has been recent technological advantages improving the PCR. There is also now a modernisation of PCR known as real-time PCR. This allows for the results to be faster and more accurate than the conventional way of testing PCR.
In 1953, James Watson, together with Francis Crick, composed a theory of DNA self-replication, suggesting DNA were able to duplicate itself using one strand of the double helix as a template, finishing with two pairs of chains as opposed to the original one. At the time of developing this theory, Crick and Watson were yet to find biochemical evidence. This theory led to two further queries: the process of unwinding the double helical structure of DNA, otherwise risking tangling everything, and if there were an enzyme involved. At this time, Kari B. Mullis was 8 years old. His adolescent years were engulfed by science, specifically chemistry, rather than biology. Mullis found interest in human biology for the first time when doing medical research. After attending a seminar describing the synthesis and cloning of the gene for somatostatin, Mullis realized that DNA could be synthesized via chemistry, and this excited him. In 1979, Mullis got a job in DNA synthesis. The lab next to Mullis's were trying to form methods in detecting point mutations. This led to Mullis asking if DNA polymerase and dideoxynucleoside triphosphates were able to extend an oligonucleotide onto a specific site on DNA. He figured he would need to raise the relative concentration of this specific site of interest. Mullis continued his thought process until it led him to considering leaving out the radioactive dideoxynucleoside triphosphates, mixed the DNA sample with oligonucleotides and added polymerase, then waited. This was the beginning of the development of the polymerase chain reaction. He spent many months talking to colleagues and his at the time girlfriend, trying to find a reason why it wouldn't have worked, or why it hadn't been done yet. He, nor anyone he spoke to, could answer these questions. Mullis first tested his theory with human DNA and the nerve growth factor primers, boiled it, followed by cooling, added about 10 units of DNA polymerase, and left it at 37 degrees Celsius. He took a 12-hour sample and found he had to heat and cool the sample whilst adding more polymerase as he went. The reason for adding more polymerase was due to the denaturation of it through heat. In 1992, a thermostable and heat-resistant DNA polymerase was derived from Thermus aquaticus, which is shortened to and known as Taq polymerase. The first generation of the polymerase chain reaction was endpoint PCR. This was invented in the 1980s for a widespread and multitude of uses, such as medical diagnosis, environmental monitoring, and food safety analysis. Endpoint PCR could only provide relative and qualitative data. The second generation of the polymerase chain reaction was real-time quantitative PCR, or qPCR, which was developed in 1992. It uses fluorescent-based probes or dyes and enables target amplification and quantitative detection. However, it isn't able to provide an absolute result and is highly dependent on operator skills. The third generation of the polymerase chain reaction is digital polymerase chain reaction, or dPCR. dPCR is a nucleic acid quantitative technique with unparalleled sensitivity, where obtaining reliable qualitative results is possible from trace amounts of nucleic acid.
So how does the polymerase chain reaction work? Well, the steps of the PCR are relatively simple. In order to replicate a DNA strand, we first need to break down the strands. The PCR reaction incorporates the tag polymerase, primers, template DNA, and the nucleotides, or the DNA building blocks. To achieve this, denaturation of the DNA strand occurs at approximately 96 degrees Celsius, and this is where the heating needs to achieve separation of the DNA strand from each other, and provides a DNA template. The annealing, or the cooling process of the reaction, requires to be approximately 50 to 65 degrees Celsius, and this allows the primers to then bind to their specific sequences within the single DNA strand template. So once the annealing or the cooling process has been achieved, the reaction needs to then go through the extension phase. The reaction is to be heated again to 72 degrees Celsius, and this will allow the tag polymers to extend the primers out and the addition of the appropriate nucleotides, resulting in the synthesizing of a new DNA strand. The cycle will then repeat 30 times in a typical polymerase chain reaction. The replication of the DNA template itself is not required from the original template, but can be replicated from the new DNA that has been synthesized. So, as you can see from the diagram in front of us, for the polymerase chain reaction to work, we have the intact double-stranded DNA templates at the top, and we need to separate these. And we go about doing this by heating the reaction to 94 degrees Celsius, and this allows for denaturation to occur, or the separation of the two DNA strands. Once this occurs, we have the annealing process that takes place, and this is where we need to cool the reaction down to 55 degrees. Doing so allows the primers to then bind to the specific sequences within the actual DNA strands. Once this has occurred, we then move into the extension phase. The extension phase allows the tag polymerase to extend the primers and for the addition of the appropriate nucleotides, resulting in the synthesizing of the new DNA strands. Once this occurs, the cycle repeats itself 30 to 40 times, and this will then allow for the new DNA template to be replicated.
There are many different ways that polymerase chain reaction is used across different scientific disciplines. Some of these include genetic research, medicine, forensic science, and environmental microbiology. Polymerase chain reaction across genetic research. Polymerase chain reaction is able to be used to assist DNA sequencing. Sections of interest from DNA are amplified to allow for the further study of genetic mutations and consequences. The Human Genome Project also used polymerase chain reaction to indicate the presence of specific genome segments in particular clones. Advanced variants of the polymerase chain reaction technique have been found useful in chromosomal analysis to assist with the early detection of genetic birth defects in children. Polymerase chain reaction is used in numerous different ways within medicine. It is used in microbiology, virology, mycology and parasitology, and dentistry. Microbiology. Polymerase chain reaction is used in microbiology for organism detection. Organisms such as Mycobacterium tuberculosis can be studied effectively with the help of genotyping. Virology. Polymerase chain reaction is used to detect and characterize the nucleic acids of viruses, which enables a greater understanding of virus behavior during infection. Polymerase chain reaction can be used to detect HIV in early stages, even prior to any antibodies being formed. Mycology and parasitology. Polymerase chain reaction can be used in early identification of microorganisms to promote efficient diagnosis and treatment of fungal and parasitic infections. Dentistry. Polymerase chain reaction is a standard diagnostic and research tool in dentistry. The polymerase chain reaction allows for the diagnosis of infectious microbes resulting in maxillofacial infections. It can also assist in the effective management of periodontal disease, endodontic infections, and oral cancer. Polymerase chain reaction in forensic science. Polymerase chain reaction is used for PCR-based DNA fingerprinting and is a vital tool in forensic investigations. DNA fingerprinting can be beneficial as tiny fragments of DNA can be isolated from crime scenes and then screened against DNA databases. DNA fingerprinting can also be used in paternity testing, where DNA from an individual is matched with possible children or any other possible relatives. The use of polymerase chain reaction in environmental microbiology. Polymerase chain reaction is used to detect and monitor waterborne microbial pathogens which may pose a major public health hazard. Applications of the polymerase chain reaction. Polymerase chain reaction is such a sensitive method of detecting microbial pathogens in clinical specimens. Due to the sensitivity, polymerase chain reaction is able to detect the smallest of changes. Where specific pathogens are problematic to culture, either due to long periods of time required, or they are difficult to culture in vitro, polymerase chain reaction becomes a highly important diagnostic tool. Whilst polymerase chain reaction can be highly important in the detection of microbial pathogens, there are a number of ways of which polymerase chain reaction may not provide accurate results. This is due to the fact that polymerase chain reaction is so sensitive that it's open to contamination and the susceptibility of PCR to inhibitors. As Yamamoto states, that the sensitivity and specificity of a PCR assay is dependent on target genes, primer sequences, PCR techniques, DNA extraction procedures, and PCR product detection methods. Polymerase chain reaction has been used as a diagnostic tool for numerous central nervous system diseases. Polymerase chain reaction has the ability to detect minuscule amounts of DNA or RNA in tissues and fluids. This has assisted in the accurate diagnosis and the enhanced understanding of the pathogenesis of some of the following listed viruses: adenovirus, dengue virus, Epstein-Barr virus, poliomyelitis virus, echovirus, human herpesvirus 6, HIV or the human immunodeficiency virus, herpes simplex virus, both types 1 and type 2, influenza virus, Japanese encephalitis virus, the mumps virus, the measles virus, Paris virus, B19, and rabies. Advantages of the polymerase chain reaction. The polymerase chain reaction technique is quite sensitive, which is beneficial when working with very small samples. This sensitivity then allows for multiple replications to occur. PCR is also able to be accurately undertaken with either exceptionally old or damaged DNA, again allowing for replication to occur. Some advantages of the polymerase chain reaction, as listed by the World Health Organization, include that it's quick, reliable, sensitive, relatively easy, and specific. Some disadvantages of the polymerase chain reaction. Whilst the sensitivity of the polymerase chain reaction is an advantage, it is also a major disadvantage, as the polymerase chain reaction doesn't require substantial pieces of DNA for replication. This may allow for small amounts of cross-contamination, which will then alter the replication of the DNA, therefore providing an inaccurate replication. Some disadvantages of the polymerase chain reaction, as listed by the World Health Organization, include the need for equipment, the cost involved, Taq polymerase is expensive, contamination, false positives, internal control, and cross-reaction. Some advantages and disadvantages of the different types of polymerase chain reaction. Standard or conventional polymerase chain reaction. Advantages. There is little cost involved. It is easy to access equipment, as most research facilities have conventional thermocyclers. Disadvantages. Qualitative results only, post-reaction handling, and low sensitivity. Real-time polymerase chain reaction. Advantages. It can generate quantitative results. The results can be sequence-specific, and there is no need for agrostel to visualize the DNA post-polymerase chain reaction, and there is increased specificity. Some disadvantages include: it is more expensive than the standard or the conventional polymerase chain reaction. The polymerase chain reaction speed is adequate, but it's not exceptionally quick. Polymerase chain reaction arrays. As some advantages include, up to 88 genes can be measured per sample at a time, with 8 control reactions for any given sample. Some disadvantages include that one array is needed per sample and it's costly for many samples. Microfluidic chip. Advantages: fast results, it's portable, small size, robust, and precise. Disadvantages: specialised equipment is required, and that is quite costly. Digital and drop digital. Advantages: it is very precise and absolute quantitation, but some disadvantages: again, it is specialised equipment which is quite costly.
The potential future applications or extensions of the PCR technique. Research into the potential use of PCR techniques has been improving over the years, and in one aspect we look at the improving of the current hospital infection control practices, which have been trialled utilising the MASCO multiplex polymerase chain reaction to confirm if several aspects of the enteropathogens are prevalent in stool samples of the patients. To this date, however, this emerged as an unrealistic option for most microbiology laboratories, without further research being conducted to explore this application in depth. A similar application was also being researched in order to detect bloodstream infections, and this was related to healthcare or lack of decent healthcare within the facilities. Utilising the reverse transcription polymerase chain reaction to detect DNA in patients' blood samples, it was found that the sensitivity levels were not effective, and therefore further research is being conducted to determine the viability of this application by improving the diagnostic sensitivity of the reverse transcription polymerase chain reaction. Relating to the actual polymerase chain reaction techniques, at present they remain expensive and technologically advanced. Future research and development of these techniques are required to make the polymerase chain reaction platforms a user-friendly diagnostic tool and improve the cost efficiency, especially for companies, in particular the rural setting hospital facilities. In conjunction with this research, polymerase chain reaction protocols require standardization and improvement in their efficiency.
For people living with cancer, some may relapse due to not all cancer cells being destroyed by a treatment. These cells are likely to continue to multiply, causing a possible chance for tumours to re-establish. The polymerase chain reaction has been used for these situations in detecting residual tumour cells where conventional techniques had implied eradication of the disease. The polymerase chain reaction has also been used to characterize other mutations. The sensitivity of the polymerase chain reaction may enable preventative measures against cancer. One possibility is to design antibodies against cancer cells.
Research and audio contributions by Elise, Andre, and Jade.
Here is the list of references we use to source our information on the use of the polymerase chain reaction in science.